and we will study how the SMARCAL1 inhibition affect the tumor genicity and response to the chemoradiation. So here's the summary. So first, the SMARCAL1 loss of function can induce the ALT pathway in glioblastoma cell line. And we have established a model system to study the role of SMARCAL1 in ALT in GBM. And by the inhibition of SMARCAL1 will increase the basal level of DNA damage in ATRX mutant ALT positive glioblastoma and offers a new approach for the ALT positive GBM therapy. So at last, I will thanks to my mentor, Dr. Haiyan, and thanks for Bill Dipas, and Matt, who we are working on the telomere project for a long time, and thanks for our lab manager, Paula, and all the other people in Dr. Haiyan's lab, and also thanks for Yiping and the members in Yiping's lab. And also thanks for the pathology PhD program. So any questions? Yeah.
Just kind of a naive question. Based on a lot of what you've shown has been a lot of in vitro data, correct?
Mm-hmm.
Is there a specific mouse model that you can actually use to look at ALT? Because I'm not really familiar, at least with a lot of the work. So.
I think for the as far as I know, there's one issue by using mouse model because the telomere of the mouse is relatively very long than humans. So I think some groups are using the telomerase defect mouse model and trying to generate some phenotype mimic the ALT. Yes.
Are there any therapeutics that appear to inhibit SMARCAL1 already? Thinking about using, or?
No, I don't think there are any inhibitor for SMARCAL1, but since SMARCAL1 is a helicase, so there's a potential to have some small molecule to inhibit this protein.
And also, if when you knock out one of these helicases, like ATRX or SMARCAL1, do you think there might be other helicases that might get recruited and step in and try to replace that function?
Well, I think that's a good question. So there are several helicases which are working on the telomere region, but based on the major function, they are helicase to unwind the G4 quadriplex, like the ATRX, and also the BLM or the WRN, these two helicase. And I think, as far as I know, SMARCAL1 is the only helicase trying to as a annealing helicase to resolve the replication fork story. So I think, yeah, definitely it's worth to test other helicase inhibition, but I think SMARCAL1 is a good target. Yes.
I guess also like as an extension of that question, like if you cultured long enough, do you think that there will be eventually other diseases to come over? Like even without like when you expose HRX and SMARCAL1, like if you culture long enough, do you think that any mutations like that you would have other cases take over?
So I think the truth is I didn't go into very detail about the APV body, and that body is a protein and DNA complex, and a lot of helicase involved in this APV body and try to resolve the replication stress. So I think it's still unclear how the helicase are cooperate to resolve this replication stress, but definitely there can be some other like helicase be recruited to the site to resolve that issue. Yes.
What is the relationship between age and these GPL tumors associated with the telomerase activity? Because as the telomerase region gets smaller as you get older, right?
Yes, I think the telomere length has no clear relationship with the age, because after the cancer cell pass the crisis and they use two different mechanisms to maintain their telomere. And study shows that the tumor cells use telomerase as the way to maintain their telomere. In that way, the telomere length is relatively short and even shorter than the normal cell. But for the other, using ALT, we are thinking that the telomere in this part of tumor has a very long telomere, which is I don't think is associated with age or yeah. Yes.
Your brief comment on the potential tissue specificity is the function of SMARCA1, because you are considering targeting. Right?
Yes.
Targeting is the question is, this protein might be important in certain tissue or or maybe every tissue. You know what I mean? Like, I was just wondering the toxicity. The mutation is pretty limited to certain tissue types, certain cancer types, right?
Yes.
So it suggests maybe that there's a
I think based on my understanding, I think the ALT tumor usually occurs either in the brain tumor or the tumor with the mesenchyma origin, like the osteosarcoma or the chondrosarcoma. So targeting the SMARCA1 can have some affection on the normal cells, but actually every cell has a threshold to tolerate the DNA damage. And for normal cell, I think even when the SMARCA1 function was inhibited, still other helicases can resolve the replication stress.
No, we're done. Okay. Bye.
Thank you.
Thank you.